Hi A&P1 students. I ran out of time on the last video. We didn't get to talk about the very last function of the membrane proteins. So I'm going to talk about in this video and then we'll jump to our next objective. So the last function that a membrane protein can have is in the cell adhesion function. This is a protein that's in the cell membrane of this cell, and this is a protein in the cell membrane of another cell. And so they can serve as cell adhesion proteins. Just means that they can connect to each other, and when they do that, then they connect two cells together. So if you think about the epithelial tissue we looked at in lab, all those cells were like tightly packed together, I should say. And so they were connected by probably by these cell adhesion proteins. And so there's cells that need to stay in place and next to each other. And so they will use cell adhesion proteins in their cell membrane to connect one cell to another. So that was the last function of our membrane proteins. So I'm going to close out of that. We're going to jump to our objectives. So we're going to start talking about membrane transport. And that is essentially just talking about how substances can move across or be transported across the cell or plasma membrane. All right. So there's quite a few ways that it can be done. So part of learning this material is just trying to remember the particular method that's used and what it means. So how things get across through these various methods. So we're going to start with the passive processes. These are the ones that there's no energy that needs to be put into this movement of a substance across the plasma membrane. So no energy is required. So they call them passive processes. And then we will talk a little bit later in another video about the ones that do require energy. But we're going to start with the ones that don't. And so generally, this is how I've organized this. Diffusion is one of the methods of membrane transport. And then there's two types of diffusion. There's simple diffusion and there's facilitated diffusion. And then of the facilitated diffusion, there's two types of it. There's channel-mediated facilitated diffusion and carrier-mediated facilitated diffusion. All right, so that's for the diffusion. And then the second type of passive process we'll talk about is osmosis. All right, so we're going to first start with simple diffusion. I'm going to go to our textbook. Okay, so first, just to help you recall what the term diffusion means, the term diffusion means when a substance moves from an area where it's in really high concentration to an area where it is in low concentration. So this image hopefully will help you visualize this. This always reminds me of coloring Easter eggs. You drop that little tablet in a bowl of vinegar or water or something, and at first when you drop it in, all the dye is concentrated. It's in high concentration in one area, and then over time, what will happen? These little molecules, these are dye molecules in here. They're constantly moving. So they're constantly moving, and what their tendency is to constantly move, and they don't like to bump into each other so much. And then even if they do bump into each other, they usually get they kind of bounce off in different directions. So they will move from this area where they're in really high concentration. You can see some of these guys moving away to an area where it's in really low concentration, which would be like down here. So this kind of shows more of them moving away from that high concentration area to the lower concentration areas. And they'll continue to do that until equilibrium is reached, which means that the concentration of all those little dye molecules is now, evenly distributed throughout this beaker of water, I'm guessing, so which is shown here. So that's the tendency of ions and molecules that are in our body. They don't want to be in an area where they're in real high concentration. If they have the chance, they're going to move to an area where they're in lower concentration. And they'll continue to move until they reach equilibrium, if they can. So, okay, so that is diffusion. So now we're going to talk, though, about this concept across the cell or plasma membrane. So we're going to talk about, let me go back to my plasma membrane here. So we're going to talk about substances moving across the plasma membrane. And again, they will want to move. So if we're talking about across the plasma membrane, we're talking about two different areas they're going to want to move between. So either move from outside the cell to inside the cell, or from inside the cell to outside the cell. And their concentration, just like in that beaker, generally for ions and molecules in the body is always different. Usually, for an example, sodium is typically always in a much higher concentration out here in the outside the cell, the extracellular area, and in a lower concentration inside the cell. And then like potassium is always higher inside the cell, its concentration, and lower outside the cell. So that's typically, you know, how those ions and molecules are in the human body. So they're generally in different concentrations on either side of the plasma or cell membrane. So if given the chance to move across the plasma or cell membrane, they're going to want to move from the area where they're in higher concentration to the area where they're in lower concentration. So if we're talking about sodium, remember, is higher outside the cell and then lower concentration inside the cell. So sodium would want to move, if given the chance, would want to move into the cell. And it would continue to move into the cell until the concentration outside equaled the concentration inside, if it was allowed to do that. So, but again, it's all, whether it gets allowed to do that, it's based on, you know, the proteins that are embedded in the plasma cell membrane and the chemical properties of whatever, in this case it's sodium. So that will all vary. Okay, so now that we have that kind of overview of diffusion, then we can talk about the difference between simple diffusion and facilitated diffusion. So let me go to our, there we go. Okay, so here's an example of simple diffusion. So whenever we have molecules that move through the phospholipid bilayer, so in this case they're using oxygen as an example. It's very small and it can move through the phospholipid bilayer. So, and you'll notice that generally in the body, oxygen is higher outside the cells than inside the cells. So oxygen will want to diffuse down its concentration gradient. That's another term they use for this. So whenever oxygen is diffusing into the cell, in this case, because this is inside the cell, and this is outside the cell, diffusing into the cell down its concentration gradient, which just means it's going from an area where it's in high concentration to an area where it's in low concentration. And then the opposite could happen if it's another ion or molecule. It may move from inside the cell to outside the cell. So carbon dioxide is an example. Generally, our cells are always making carbon dioxide as a waste product, so it's generally always higher inside our cells. And then there's less carbon dioxide outside our cells. So, and carbon dioxide is one of those small nonpolar substances like oxygen, so it can easily kind of go diffuse through that phospholipid bilayer and go from an area of high concentration to low concentration. So that's simple diffusion. So we got these molecules that are moving through the phospholipid bilayer without any help, and they're diffusing, which means they're going from an area of high concentration to low concentration. All right. And then facilitated diffusion. This is when the facilitated diffusion is when the molecules or ions can't move through that phospholipid bilayer. They need help to do it. So they may be like a little bit larger, or they may have a charge on them. So they could be like a polar molecule. They could be an ion. Those are things that have charges on them. So they can't get through that phospholipid bilayer. So they need some help to do that. And so whenever they need help, the plasma membrane proteins, remember that was one of the functions of some of those proteins in the plasma membrane was to transport things across. That's where they will be helping out some of these molecules that can't go through that phospholipid bilayer. So in our picture here, sodium is an example. It has a charge on it. It has that +1 charge on it. So it's kind of repelled and not able to pass through the phospholipid bilayer. But if there's a protein present, remember the proteins have a specific shape, and this is a sodium protein channel, so it only lets sodium. It's very specific for just sodium, so it'll only let sodium in. And then you'll notice that the sodium typically in the body, is always in a higher concentration outside the cell than inside the cell. So sodium will diffuse down its concentration gradient, which just means it'll go from a higher concentration area to a lower concentration area, and it'll diffuse into the cell. And then potassium, it also has a charge, so it's going to need help getting across. So they call this type of facilitated diffusion, remember there's 2 types of them. They call it channel mediated because this is just serving as a channel. It's like a protein with a channel right down the middle of it. They kind of have it the other half cut off so we can see the sodium moving through here and the potassium moving through. But this is like just a protein that has like a little channel going right down the center of it. And so anytime the sodium wants to move from an area of high concentration to low concentration, it'll do that, and the potassium same way. So they call that channel mediated facilitated diffusion because the particular substance is moving across the plasma membrane using a channel. All right, and then the other type of facilitated diffusion is carrier mediated facilitated diffusion, and that just means that a substance is still using a protein to go from an area where it's in high concentration to an area where it's in low concentration. So in this case, they're using glucose. Glucose is typically higher outside the cells than inside the cells. So it'll want to move down its concentration gradient. So move from this area of high concentration to this area of low concentration. And it's facilitated because it'll use a protein. But the only difference is they call it carrier mediated because this protein actually, and this is kind of showing the same protein, it's just when the substance binds, the protein changes shape. So you can kind of see how it was this shape and now it's this shape. And then it changes shape again to carry that glucose molecule over to the inside of the cell, across that membrane. So they call that carrier mediated. So these are both facilitated diffusion because they require membrane transport proteins to get across. This one uses a membrane protein that has a channel in it, and then this one uses a membrane protein that changes shape to get the substance across. So, all right. So that is our simple diffusion and facilitated diffusion. So in the next, and these are mainly talking about substances like sodium and oxygen and glucose. The next thing we'll talk about is osmosis, and this just has to do with the movement of water across the cell or plasma membrane. So that'll be in our next video.